Again, I'm back. That video was like a little longer, so I'm gonna try to make this quicker. I think I talked too much. But anyway, let's get back to where we were. So what is a chromosome? A chromosome is just a chromosome is made up of DNA tightly packed or coiled many times around proteins called histones. So if you see the word histones, histones are just proteins that are tight, that are they just kind of help to be tighter, to be just kind of coiled around DNA. This is what it is. Then we have, what is the difference between phenotype and genotype? So the phenotype is just kind of all of the observable characteristics which are influenced by its genotype and environment. So genotype is the set of genes that an organism carries. So if you're comparing this to a phenotype, it's just really like what you can visibly see in someone, whereas the genotype is like the genetics within you and that you just cannot see at all because they're not visible unless you're like a clear, transparent person, which you're not. Next we have, what is an example of a genotype? So we have a genotype, for example, could be used letters, like capital letters, one capital A, one capital A, one capital A, and one lowercase a, or two lowercase a's. For example, you can also use like homozygous, dominant recessive, or heterozygous. It all depends on what we're talking about. Remember that we have, at the end, we're gonna have 3 different, we'll talk about this later, I'm not gonna get into that right now. But we also have, what is an example of a phenotype then? So this could be purple flowers, white flowers, blue eyes, brown hair, dark skin. Anything that you can visibly, physically see. Okay. So what is incomplete dominance? Incomplete dominance is a cross between organisms with 2 different phenotypes which produces an offspring with a 3rd phenotype that is blending the parental traits. So if you need an example for what incomplete dominance is, remember when you mix, God, that's very sunny. Remember when we mix a white flower and a red flower, we get pink flowers because there's a mix of both phenotypes, and thus you get another 3rd phenotype with a mix of these 2. So remember, this is just a mix of both of the parents. Just in essence, that's what it is. Now, if we talk about codominance, we see that codominance is both traits appear together. So you're not going to see a blend or mix, you will see both traits appear in that offspring. So for example, if we have a white cow mixing with a, let's use a chicken example. We have a black chicken and we have a white chicken. You're going to see a checkered chicken. So you'll see both colors be present in the chicken. Perhaps as spots or whatever it is. You will not see a blend of 2 colors, you will see both colors. Just remember, that's the difference between codominance and incomplete dominance. Now we have, what does homozygous mean? So homozygous is just a trait that's made up of 2 of the same alleles. So it can either be dominant or recessive. Again, homozygous is like capital A, or capital or lowercase a, lowercase a. Then we have, let's see, I have a problem here that I did with like the Punnett squares, which you should be practicing because they will be on your exam for sure. But I will get into that later. So what are the base pairing rules in RNA? So we have adenine and uracil and guanine and cytosine. What are the 6 types of chromosomal mutations? So we have translocation, deletion, duplication, inversion, nondisjunction, and insertion. I think you might want to look at what each one of these mean. Again, chromosomal mutations, as we talked about, these are the ones affected by your environmental factors and such as ultraviolet radiation, in what I'm receiving right now, but that's just what it all is about. So what happens during deletion? So during deletion, one of the letters is just like completely taken out of. I will put a picture right here so you can better understand it, because I'm a very visual learner, so that helps me out quite a lot, but so you can better see what I'm talking about. Deletion is just like taking out a letter completely. Next we have, I'm sorry, I'm struggling, I know. So what happens during inversion? So inversion, as you can see, like inverting things. Just think of like the root of it. That might like give you a hint. But inversion is like when 2 letters are inversed in places of 1. So if we have like A, B, C, D, E, F, then the outcome is going to be A, B, E, D, C, F. So the 2 letters just kind of switch places. Again, I'm going to put a picture so you can understand what I'm talking about, because I know I probably am not making any sense at all, and that's okay, because I'm a very bad, whatever. So what is the difference between a mutation that occurs during mitosis and one that occurs in meiosis? This is a very tricky question, not so hard to understand, but in mitosis, the mutations happen early or late in development. Whereas in meiosis, sperm or egg cells give rise to egg and sperm. Okay, that probably made no sense. The mutation will be incorporated into the gametes and passed on to the offspring. That is kind of the last thing I said was for meiosis. Again, that doesn't make enough sense, please look this question up. You might find it somewhere. It's really important to know because I'm sure it's going to appear in the exam. Next question: during what stage of mitosis and meiosis do the chromosomes separate? So these chromosomes again separate during anaphase when the spindle pulls them apart. That's just very important to know. So when chromosomes fail to separate, what is this called? So this is called Nondisjunction. Remember, these are the 6 chromosomal mutations that I talked about. This is one of them, Nondisjunction, is when the chromosomes fail to separate. So what is different between how chromosomes separate in mitosis versus how they separate in meiosis? So in mitosis, I'm going to show you pictures of this. I think it's easier this way. But in mitosis and meiosis, you will see like the way the chromosomes line up in the middle is different, and that's how they separate. Because remember, in meiosis, you have to go through 2 cell divisions. You have meiosis 1 and meiosis 2. So just remember that. If you have any questions, please go ahead and email me or just like leave a comment, whatever. I will get back to you as soon as I can, because I really don't know what I'm doing now. But thank you guys for watching, and I hope you do well in your exam. Please study and don't procrastinate.